Move on to DNA repair. So as we all know, maintaining the integrity of DNA information is absolutely essential to life and is supported by an elaborate system of DNA repair mechanisms. And so if we look at DNA, it can be damaged spontaneously over time or in response to environmental agents, and actually replication itself can damage DNA. So chemical reactions and some physical processes are constantly assaulting or damaging genomic DNA, and a majority of these are corrected using undamaged strand as a template. So some base changes can escape repair, and in that case an incorrect base serves as a template in replication. And that results in a daughter DNA that carries the changed sequence in both strands and ultimately results in what we call a mutation. Accumulation of mutations in eukaryotic cells is strongly correlated with cancer, and we know that most carcinogens are also mutagens. When we look at DNA replication and DNA metabolism, there are thousands of lesions that occur every day. But ultimately, only 1 in about 1,000 become a mutation, and that's because of the successful repair through these DNA repair mechanisms. When we look at the human genome, it contains genes for more than 130 repair proteins. This large number of proteins dedicated to a process really highlights its importance. So first we need to get into the vocabulary of DNA lesions. So first, a lesion is basically any DNA damage. If unrepaired, this lesion becomes a mutation. Mutations can be substitutions or point mutations. They can also be deletions and additions of nucleic acid sequence. A silent mutation has no effect on gene function, or it affects a non-essential region of the DNA. In contrast, there are non-silent DNA mutations which do result in a change in the amino acid sequence. And most of these non-silent DNA mutations are neutral or deleterious. Very rarely are they advantageous. So the Ames test was developed by Bruce Ames, and it's used to identify or determine the mutagenic potential of a compound. So this assay or test uses Salmonella strain. This strain has a mutation that makes the bacteria unable to synthesize histidine. And so if you plate these cells on a histidine minus plate, they will not grow. Basically, in the assay, you add a compound to that plate of Salmonella and look to see whether or not it grows in histidine-free media. So the presence of colonies indicates that the compound has mutated the Salmonella and restored its ability to synthesize histidine. And on this slide, we see an example of what results from an Ames test would look like. And in panel A, this is basically the Salmonella that are plated with no addition of the potential mutagen. And what we see here is that there are some colonies that do grow, and this is because of spontaneous mutation. So if we add a mutating agent, we would expect the number of colonies to increase. In panels or in plates B through C, what we see is that each of these discs have been incubated with fewer and fewer amounts of the carcinogen, or lower amounts of the carcinogen. And what we see here is in the highest concentration you get a cell-free zone, and that's because at very high concentrations of the mutagen, it's lethal to the cells. And what you see is as you look at the area further away from that disc, you see that you do start to get appearance of colonies that are able to grow. Under in the presence of lower concentrations of the mutagen, you now see that you get large numbers of colonies that are now able to grow. And so basically this indicates that the mutagen, that the potential substance, or that the substance that you're looking at is indeed a mutagen. When we look at all of the currently known carcinogens, about 90% of them are mutagenic in the Ames test, and so it seems to have it's a fairly robust assay. Now it's not perfect, so there are compounds that would be positive in the Ames test that that are not carcinogens, and then some carcinogens that do not look mutagenic using the Ames test. But it's used because it's cheap and it's easy. And it's easy to adapt to a high throughput assay. Now sometimes compounds are not mutagenic until they're processed by the cell. And one kind of adaptation of the Ames test is that you can incubate the compound with liver extract. And in that incubation, the mutagen can be activated to a more mutagenic form of the compound. So a lot of times that extra step has been added. So there are several types of DNA damage that we'll talk about. The first is mismatch. So mismatches arrive from occasional incorporation of incorrect nucleotides. There are also, in DNA damage, we can see abnormal bases. These arise from spontaneous deamination, chemical alkylation, or exposure to free radicals. So basically chemical alterations of the DNA. We also can observe pyrimidine dimers, and these form when DNA is exposed to UV light. And there are also backbone lesions, and these typically occur from exposure to ionizing radiation and free radicals. And the next slide, in table 25.5, we see enzymes and proteins that are involved in DNA repair. And in the first column, you see the enzymes and the proteins. So here we see the different proteins that are involved in mismatch repair, proteins that are involved in base excision repair, nucleotide excision repair, and direct repair. And those are the processes that we'll talk about. And then these are the types of damage that each protein is responsible for correcting. So mismatch pair relies on methylation. And so once an incorrect base or nucleotide has been incorporated into the DNA, the cell needs to then be able to identify which is the correct mutation, or which is the correct sequence and which is the incorrect one. And the way it does this is through methylation. So in E. coli, the parent strand of the DNA is mutated, is methylated. And this methylation occurs through the activity of a DAM methylase. And basically, this enzyme inserts methyl groups at adenines in the GATC sequence. So following a short time after the daughter strand is synthesized, it becomes methylated by this DAM methylase. And so immediately after DNA replication, that daughter strand is unmethylated, and that is how the cell can identify the correct nucleotide from the incorrect. So in methylation of DNA and repair, the newly synthesized strand is unmethylated for a short period after synthesis. So replication errors would then reside in that unmethylated strand. So the methyl-directed mismatch repair system is able to cleave the unmethylated strand in the initial part of the repair process. And so here is a figure of the mismatch repair and methylation process. So here we see that the parental strand is fully methylated, so it has a methyl group on both strands. After replication, we see that the daughter strand is not methylated, and again, the parent strand is. So after this initial round of replication, each DNA is hemimethylated. Now, after a few minutes, the new strand is methylated, and these can no longer, the parental and the daughter strand can no longer be distinguished from each other. And so the repair has to occur at the point in time at which the DNA is hemimethylated. So the proteins that are involved in the recognition and repair in E. coli, first there is the mut-L and the mut-S proteins. These recognize that methylated GATC. Then mut-H binds to that mut-L, mut-S DNA complex and forms a DNA loop. This DNA then strands through the complex until the methylated GATC is encountered. And this mismatch is often up to 1,000 base pairs away from that methylated GATC. So the mut-H cleaves the non-methylated DNA strand on the 5' side of that G. The DNA unwinds and is then degraded in a 3' to 5' direction. Helicase 2, also referred to as UVRD helicase, a single-stranded binding protein, and several exonucleases work to degrade the non-methylated DNA towards the mismatch, and that removed sequence is replaced using a DNA Pol III and a DNA ligase. So here we see the early steps of methylation-directed mismatch repair. So in this initial panel, what we see is the parental correct base pair is methylated, and here and designated here is the mismatch base pair. If we look at the actual sequence, we see the GATC. And again, the blue parental strand is methylated and the daughter strand is not. So at the site of the mismatched base pair, the mut-L, mut-S complex forms. And this binding is facilitated or accompanied by ATP hydrolysis. And the next step, so here we see the mut-L, mut-S complex is formed. Now mut-H comes in and mut-H binds at that complex, and the DNA is threaded through. And so it proceeds bidirectionally. After the formation of this complex, the mut-H cleaves that unmethylated strand. And so we see here the daughter strand is cleaved. And the next thing that has to happen is that this nucleic acid sequence containing the incorrect base pair must be degraded. And so we see that this degradation can occur through one of two mechanisms. And the mechanism that's taken depends on where that daughter strand has been cleaved. So in the left-hand side, we see the process that occurs if that daughter strand is cleaved upstream or at the 5' end of from the sequence. And then in the right-hand side, we see what happens when the cleavage occurs 3' or downstream of the mutation. So if the cleavage is 5', there is again the mut-S, mut-L complex works with DNA helicase 2 to unwind the complex or the DNA, and then exonuclease 7 or REC-J nuclease are then involved in the removal of that sequence. So DNA polymerase 3 and single-stranded binding protein then work together to synthesize the correct strand, and as we have seen before, DNA ligase then comes in to seal the nicks. Alternatively, if the sequence is cleaved at the 3' end, we still have the mut-L, mut-S complex and the DNA helicase, but now we have two different exonucleases, or the potential of 3 different exonucleases that then work to degrade this nucleic acid sequence. Again, in the final step is the same as the first process, in that DNA polymerase III and the single-stranded binding protein work together to fill in this nucleic acid sequence. And again, DNA ligase comes in to connect the two. So when we look at the overall process of DNA methylation and repair, ultimately what happens is that the cleaved strand with that incorrect nucleotide is degraded by exonuclease. It is then rebuilt by DNA polymerase using the correct template strand, or the correct parent strand as template, and it is sealed with DNA ligase. Now, a thing to keep in mind is that ultimately this repair is quite costly. So for every single nucleotide mutation, when you correct one nucleotide, you degrade 1,000 nucleotides. So this is a lot of energy that the cell is expending to maintain that genomic integrity, and that really highlights or accentuates why this genomic integrity is so important. So another form of DNA repair is base excision repair, and this process uses a specific family of proteins called DNA glycosylases. Each DNA glycosylase recognizes specific lesions. So these enzymes then cleave the N-glycosyl bond that links the sugar to the base, and this results in an apurinic or apyridimic stream, AP site. Uracil glycosylase removes uracil from DNA. This is important because C spontaneously deanimates to U, and we all know that U doesn't belong in DNA. If we look at cells that lack this enzyme, they have a very high rate of GC to AT mutation. There are other glycosylases that make AP sites at different lesions. So there's 8-hydroxy-G, hypoxanthine, 3-methyladenine, and so forth. So each enzyme is responsible for recognizing a different defect. So if we look at repair of AP sites in bacteria, it's interesting to note that that entire nucleotide is ultimately removed, and it's not just that damaged base. In fact, sometimes the region around that AP site is removed. So the repair is not made simply by inserting a new base and reforming the glycosidic linkage. So what happens is that these AP endonucleases cut the DNA backbone around that AP site and remove the DNA. DNA is then synthesized by DNA Pol 1, and then DNA ligase seals the nick. And so this process is represented here. So here we see a DNA glycosylase. In this case, it's a uracil glycosylase. And so it cleaves that N-glycosyl bond, releasing that base. Here you see the AP site. Then the AP endonuclease comes and cleaves the DNA at that AP site. DNA polymerase I then comes in to synthesize the new strand using the correct strand as template, and then DNA ligase finally comes in to seal the pieces of DNA. So DNA lesions that result in large distortions of helical structure of DNA are repaired by nucleotide excision pathway. These lesions include pyrimidine dimers, which are formed from UV light. Also, there are 6,4 photoproducts, which are also formed from UV light. And then benzopyrenoguanine is formed from cigarette smoke. So those are just some of the lesions that are formed and recognized by enzymes in this pathway. This pathway involves removal of a DNA segment by large protein complexes called exonucleases. These exonucleases are multi-subunit enzymes. So the nucleotide excision repair pathway in bacteria uses the ABC exonuclease. These exonucleases cleave the backbone in 2 places. When we see one example of an exonuclease is ABC exonuclease. This contains UVRA, UVRB, and UVRC. This complex hydrolyzes the 5th bond on the 3' side of the lesion and the 8th bond on the 5' side of the lesion. Typically, these enzyme complex will remove 12 to 13 nucleotides, depending on whether a single or more than one mutation is present. And again, as we've seen multiple times, DNA polymerase I and DNA ligase then come in to replace the DNA and seal that gap. So nucleotide excision repair also occurs in eukaryotes. In this example, or in this family of organisms, the enzymes hydrolyze the bond on the 3' end and the 22nd bond on the 5' side of the lesion, and it removes a larger sequence. So in this case, it removes 27 to 29 nucleotides. And then the gap is filled using DNA polymerase epsilon and DNA ligase. So the general pathway of nucleic excision repair is similar in all organisms. So in this first step, the exonuclease binds to the DNA at the site of a bulky lesion, and it cleaves the damaged DNA on either side of the lesion. So here's the lesion. It can cleave upstream or downstream. A DNA helicase then comes in and assists in the removal of that nucleic acid sequence that contains the lesion. DNA polymerase I or DNA polymerase epsilon then comes in to synthesize the correct sequence using the non-mutated strand as template, and then DNA ligase comes in ultimately to seal the nick. So another form of DNA repair is direct repair. One example of this are photolyases. These are enzymes that use light energy to repair pyrimidine dimers. Interestingly enough, these enzymes are not found in humans or placental mammals. An additional pathway of direct repair involves O6-methylguanine DNA methyltransferase, and this repairs methylated guanine. O-methylguanine is a common and highly mutagenic lesion that needs to be repaired. There is also another pathway of direct repair that involves ALK-B. This enzyme demethylates 1-methyladenine and 3-methylcytosine. And this is important because amino groups of adenine and cytosine are often methylated. And this methylation results in incorrect base pairing. So these methylated nucleotides need to be corrected, or mutations will be incorporated into the genome. So in this slide, we see how O6 methylation leads to mutation. Here we see the correct base pair between guanine and cytosine. And with O-methylguanine, what happens is this methyl group interferes with the ability to form this hydrogen bond between cytosine and guanine. And what happens now is this methylation results in incorrect base pairing. So where you normally see a G to C, this O-methylguanine wants to pair with thymine. Once this happens, once that incorrect base pair is incorporated, it becomes integrated into the genome. And here we see ultimately we have this GC base pair, the methylated GC base pair. Now what happens is that methylated G incorrectly base pairs with a T. And here we see a GT base pair. And what's going to happen is that that is going to get maintained in the genome. And so now what we see ultimately is a GC to AT mutation. So all of these repair mechanisms use the undamaged DNA as a template to correct mutation. However, there's not always, that doesn't always occur. So sometimes the template strand itself is damaged. And we see this with double-stranded breaks, DNA cross-links, or when there's damage to single-stranded DNA. All of these lesions or unrepaired lesions tend to cause the replication fork to stall. Once the replication fork has stalled, DNA can be repaired using either another chromosome as template, this results in recombination, or through error-prone translesion DNA synthesis, which we'll call TLS. So if the replication fork encounters an unrepaired lesion or strand break, replication generally halts and this fork may collapse. What happens then is that a lesion is left behind in the unreplicated single-stranded segment of the DNA. And right here what we see is that single-stranded DNA doesn't get synthesized, and that results in recombinational DNA repair or error-prone repair. Alternatively, an unrepaired break may result in a double-stranded break. In the case of a double-stranded break, there is recombinational DNA repair. So in this case, another chromosome has to serve as a template. So error-prone TLS in bacteria. So again, this TLS is translesion DNA synthesis. This TLS is basically part of the SOS response, and this is what happens when response or when DNA damage is extensive. And so SOS proteins include UVAR and UVRB, and then also two proteins called UMUC and UMUD. UMU was given that name because the strains were unmutable, and basically removal of those UMU genes abolished that pathway. So in the pathway, UMUD prime is cleaved, and this protein, along with UMCC, binds to another protein REC-A, and this creates DNA polymerase 5. And this polymerase can process past that damaged area, but what happens is that this is an error-prone complex, so proper base pairing is often impossible in such a system, and these results and this process results in a large number of errors, which ultimately lead to mutation. And we've spent a lot of time talking about how important it is to correctly repair your DNA. So the question is, why do cells have error-prone repair? And one way to think about this is that it's a desperate last-ditch attempt to live, and that's why we call it the SOS system. And ultimately, it kills some cells and also mutates DNA in the cells that survive. And it's the survival of these cells that really is the driving force for this process. So some of the daughter cells that have the mutation are able to survive. So TLS polymerases abound in mammals. So most recognize a very specific type of damage, and they have an appropriate response. One example of this is DNA Pol Eta, and this is a DNA that assists when we have those thymine dimers. The thymine dimers then cause a pause or a halt at the replication fork. And most TLS polymerases in eukaryotes are limited to very short regions of DNA, and this helps to minimize their mutagenic potential. So we are not going to have time to go into recombination. So just to summarize what we've discussed in chapter 25, we've learned that DNA replication is catalyzed by DNA polymerases. They use one of the strands as template while adding new nucleotides to the 3' end of the newly synthesized chain. We've talked about several mechanisms that exist to correct mismatches and other changes in DNA. And during DNA repair, most of the time the information encoded in the daughter strand or in the parent strand can be used to make corrections in the daughter strand. And then we've looked at this TLS system that is used to correct error.